Hello students, this is Dr. Tikia Arashiro. In the previous podcast, we talked about the SEC translocase system. Today's podcast will focus on the TAT translocase system and the different extracellular section pathways in prokaryotes. Prokaryotes translocate or export specific proteins into or through membranes. For example, the SEC translocase system or the general secretion pathway both export unfolded proteins into or across the plasma membrane. By contrast, the TAT translocase system transports previously folded proteins into or across the plasma membrane. Both of these systems are universal in bacteria and archaea. So the TAT system is dedicated for transport of folded proteins in or across the plasma membrane. Just like the SEC system, the TAT system is located in the plasma membrane. The system is called TAT because the transported proteins contain a short signal with a pair of arginine residues. TAT actually stands for twin arginine translocation. The TAT translocase consists of three membrane integrated subunits, TAT-A, TAT-B, and TAT-C, that together form a receptor and a protein conducting machinery for the TAT substrates. TAT-A is a channel that mediates the actual translocation event. It is similar to the SEC-YEG. TAT-B and TAT-C form a complex, and this complex is involved in recognition of TAT signal peptide. The complex also carries folded protein to the TAT-A. SPase1 is a signal peptidase. It cleaves the leader sequence of the signal peptide. Now, the signal peptide consists of a pair of arginine residues, serine, arginine, arginine X, phenylalanine, leucine, lysine. The energy for translocation is provided by the proton motive force. Now, the TAT system translocates folded proteins across biological membranes, and therefore it is believed that its physiological role is to extend the set of translocatable substrate to those that fold prior to translocation. This is the fundamental functional difference to the SEC system, which only translocates largely unfolded proteins. Some proteins must be folded before they are translocated because they contain cofactors or coenzymes as the protein folds. These proteins include redox proteins that utilize the following cofactors and coenzymes. Now, these redox proteins include electron carriers like iron sulfur proteins and cytochromes, and respiratory enzymes such as nitrate reductase. So these are normally folded before they are translocated or exported via the TAT system. So the TAT system is necessary for a multitude of redox pathways, such as anaerobic respirations with trimethylene N-oxide, dimethyl sulfoxide, arsenate, or chlorinated unsaturated compounds. The TAT system is also indispensable for the global nitrogen cycle, as nitrous oxide reduction depends on the TAT system. Now, anaerobic respiratory or photosynthetic pathways, which involve the cytochrome BC1 complex, also relies on the TAT system because cytochrome is a cofactor containing TAT substrate. Many bacteria possess a twin arginine translocation or TAT pathway that exports folded proteins into or through the cytoplasm. The peptides exported by the TAT pathway possess N-terminal signal sequences with a twin arginine RR motif. Prior to export, the TAT peptide is folded. The folded pre-protein, which contains an N-terminal signal sequence shown here in black oval with a twin arginine RR motif, is recognized by the TAT-B and TAT-C complex. TAT-B and TAT-C then deliver the peptide to TAT-A. Now, TAT-A is a channel through which the peptide translocates across the membrane. Now, before it translocates, the signal sequence is removed by signal peptidase. Now, I want to talk about extracellular proteins. Extracellular proteins are proteins that are secreted outside the cell. These proteins include hydrolytic enzymes like proteases, nucleases, lipases, and amylases, toxins like cholera toxin, and structural proteins like pillin and flagellar proteins. These proteins are secreted through a variety of protein secretion systems. So how are extracellular proteins transported outside the cell? In gram-positive bacteria, the unfolded proteins use the seq system and the folded proteins use the TAT system. So the peptides use either the seq system or the TAT system to cross the plasma membrane. Then they pass the periplasm and the peptidoglycan to get into the extracellular space. It is that simple. Now, it is more complicated for the gram-negative bacteria. Gram-negative bacteria have more hurdles because the peptides not only have to cross the inner or plasma membrane, but also have to cross the outer membrane to be translocated on the surface or into the extracellular space. Because of this, gram-negative bacteria use special secretion systems to transfer proteins or effector molecules across the membranes. These secretion systems are known as types 1 through type 6 secretion systems. Now, some gram-positive bacteria and archaea also possess secretion systems similar to some of these, but in these organisms, the machinery only has to transport the proteins or effector molecules across the plasma membrane. Types 1 to type 6 secretion systems facilitate several cellular activities, including symbiotic interactions, biofilm formation, extracellular enzyme secretion, transfer of DNA, release of antibiotics, and delivery of proteins into host cells. Thus, molecules secreted by these systems allow bacteria to interact with the environment and other organisms. Each of the systems specifically recognizes its substrate based on amino acid residues and is composed of a large complex of proteins that forms a translocase channel spanning one or more membranes through which the secreted molecule travels. So let's talk about the type 1 secretion pathway. The type 1 secretion pathway is a seq-independent pathway, meaning it does not depend on the seq pathway to first transport the peptide across the plasma membrane. The secretion apparatus consists of 3 secretory proteins. 2 are inner membrane proteins, and 1 is an outer membrane protein. One of the inner membrane protein is an ATP binding protein that belongs to the ABC exporters or ATP binding cassette superfamily of proteins, which also catalyze extracellular polysaccharide export, solute uptake, and drug export. The fact that the ABC transporter has ATP binding sites indicates that ATP binding and/or hydrolysis drives the secretion of the proteins. Now, the other 2 secretory proteins are called auxiliary proteins. One of these is anchored in the inner membrane or plasma membrane and has a periplasmic domain that is believed to connect to the other auxiliary protein and to the outer membrane protein. Peptide does not require to have a leader sequence and a chaperone to be transported to the membrane. It is directly secreted from the cytoplasm to the outside of the cell without entering the periplasm due to a secretion apparatus HLYD that spans the periplasm. Examples of proteins secreted by the type 1 system include alpha-hemolysin, which is a toxin that lyses red blood cells and kills other cell types, proteases, and leukotoxin, which damages white blood cells. Now I want to talk about the type 3 secretion pathway. Type C secretion pathway is a seq-independent system. The system forms an apparatus used for the injection of virulence proteins directly into the cytosol of eukaryotic host cells. These virulence factors include toxins, phagocytosis inhibitors, promoters of cell host suicide, and stimulators of cytoskeleton reorganization in the host cell. Because it is a sec-independent pathway, the peptides are directly secreted from the cytoplasm to the outside of the cell without entering the periplasm due to a channel that spans the periplasm. The peptide does not require to have a leader sequence. However, the transport process is assisted by a cytoplasmic chaperone called zinc chaperone. It is an ATP-dependent process. Now, YSCN here is an ATP hydrolyzing protein that drives the secretion of the proteins. Now I'm going to talk about the type 2 secretion pathway. The type 2 secretion pathway appears to be the major route by which proteins are secreted by gram-negative bacteria, especially hydrolytic enzymes and toxins. The production of type 4 pili, which are responsible for adhesion and twitching motility, is also dependent upon the type 2 pathway. This pathway is present in a number of plant and animal pathogens like Ehrlichia carotorova, Klebsiella pneumoniae, Pseudomonas aeruginosa, and Vibrio cholerae. It is a sec-dependent pathway. The peptide to be transported has a leader sequence and is unfolded. The transport process proceeds in two stages. Stage 1 uses the sec pathway or the general secretory pathway. In this process, the unfolded protein is transported via the sec system in the periplasm. Once the protein has crossed the plasma membrane, the leader sequence is proteotically removed on the external face of the inner membrane by the signal peptide LSPA, and the protein then enters the periplasm for the second step. In stage 2, the protein is secreted from the periplasm across the outer membrane to the external medium. The translocation machinery and mechanism of the second stage are not well understood. The secretory apparatus for the second stage of secretion is called the secreton, and it consists of 12 to 16 proteins. Not all the proteins are shown here. Surprisingly, the proteins of the secreton are located in both the plasma and the outer membrane, with most being located in the plasma membrane. Several of the plasma membrane secreton proteins have extensive periplasmic domains. Now, pool D proteins are called secretins, and these are outer membrane channels that translocates the protein outside the cell. So let's talk about the type 5 secretion pathway. The type 5 secretion pathway is structurally the simplest protein secretion system. It relies on sec pathway to move proteins across the plasma membrane. The type 5 secretion pathway is also called autotransport pathway because the proteins transport themselves across the outer membrane without an aid of an obvious secretion system. The secreted proteins referred to as autotransporters include proteases, toxins, and adhesins. The type 5 pathway is widespread among the gram-negative bacteria. The translocation uses the sec pathway to move the autotransporter through the plasma membrane into the periplasm. But in contrast to the type 2 system, there is no requirement for a separate secretion apparatus to move the protein from the periplasm through the outer membrane to the outside. So how is this done? First, let's take a look at the autotransporter. The autotransporter has three domains: an amino domain that is recognized by the sex system, an internal domain called the passenger domain, and a carboxyl terminal domain called the helper domain. The C-terminal helper domain inserts into the outer membrane to form a secretion channels to which the passenger domain is exported into the cell surface. So the helper domain is able to form a channel in the outer membrane through which the protein transports itself. The protein crosses the outer membrane without the aid of an obvious outer membrane channel. So that's the type 5 secretion pathway. Moving on to the type 4 secretion pathway, the type 4 secretion pathways are the most ubiquitous pathways and are present in many bacteria and archaea. It is a unique type of protein secretion pathway because it translocates not only proteins but also DNA across the plasma membrane and the outer membranes of gram-negative bacteria. While this system can be used to transport toxins into host cells, its primary role is to transfer DNA to other cells through the process of conjugation, which is a mechanism that prokaryotic cells have to exchange genes. Now, the secretion pathway uses either the Sec-dependent pathway or the Sec-independent pathway. When translocating protein across the plasma and outer membranes, the cells use the Sec-dependent pathway. An example of a protein translocated using type 4 secretion pathway is the pertussis toxin produced by Bordetella pertussis, which causes the respiratory tract infection whooping cough. Now, the protein translocation process occurs in two stages. In the first stage, the pertussis toxin translocates itself across the plasma membrane and into the periplasm using the Sec pathway. In stage 2, pertussis toxin enters the translocation apparatus that spans the plasma membrane, periplasm, and outer membrane and is secreted into the extracellular medium, where it is taken up by cells of the upper respiratory tract, causing some of the symptoms of whooping cough. Now, the DNA translocation process is Sec-independent, meaning it does not rely on the Sec pathway. The DNA is translocated using the type 4 translocase in one step. Here is the type 4 translocase, and as you can see here, it is similar to a pilus that extends through the outer membrane into another cell during conjugation. In this translocation process, the DNA is transported across the plasma membrane and the outer membrane via this translocation apparatus, which is connected to the pilus. Now, once the tip of the pilus makes contact with a receptor on the host cell, the DNA is recognized by an inner membrane protein of the donor and then transferred using ATP-facilitated transport. Not only can DNA be transferred from one bacterial cell to another using a type 4 system, DNA from the plant pathogenic bacterium Agrobacterium can be transferred to host plant cells through the system encoded on the TI plasmid. The TI plasmid is a tumor-inducing plasmid present in pathogenic bacterium Agrobacterium tumefaciens. The plasmid has a tDNA region that contains genes involved in tumor formation in trees, such as oak trees. The tDNA is transferred from the bacterium into the host plant's nuclear DNA genome via the type 4 secretion system. Now let's talk about the type 6 protein secretion pathway. The type 6 protein secretion pathway is widely distributed in gram-negative bacteria, and like type 4 systems, they are capable of delivering a diversity of proteins directly into the cytoplasm of other cells using a one-step ATP-requiring process similar to type 3 and type 4 systems. However, unlike the pilus-like structure of the type 3 and type 4 translocase systems, the type 6 translocase is cytoplasmic and forms a needle-like protein with a pore-forming protein that contracts all the way through the donor cells to membranes and directly into a host cell once a substrate molecule is recognized. The overall delivery process is similar to the mechanism that the tailed bacteriophage T4 uses to deliver DNA into an E. coli cell through tail contraction. Type 6 systems are used by bacteria as weapons to compete with other bacterial cells or to attack eukaryotic cells. In Pseudomonas aeruginosa, a type 6 system is used to inject a toxin into competing bacterial cells. Similarly, Vibrio cholerae uses its type 4 system to inject enzymes into competing bacteria. The enzymes degrade the cell wall and the membrane. Vibrio cholerae also uses the system to deliver toxins to human intestinal cells during a cholera infection. It should be obvious by now that protein secretion is a crucial aspect of the biology of bacteria and archaea, and that these cells have evolved diverse structures and unique mechanisms to deal with protein secretion. However, these elaborate systems are of interest to more than just basic science. Since many of these systems are essential virulence factors for the pathogenic bacteria that produce them, they are currently targets for vaccine development. The rationale here is quite simple. If the activity of the systems could be blocked by the immune system, the pathogen would be unable to establish an infection and cause disease. So that concludes this podcast on protein transport and secretion. Thank you for your attention. I hope you enjoyed watching and listening to this podcast. If you want to read more about this topic, you can refer to these sections from Chapter 18 of the recommended text, The Physiology and Biochemistry of Prokaryotes.